I'm going to dissect a cow eye. This particular cow eye is a fresh eye, meaning it has not been preserved. So a lot of times for dissections you'll get a preserved eye. It's been preserved in formalin and then soaked in alcohol. And that works great because then the tissue doesn't rot and break down as easily, but it also is much harder and stiffer. So with a fresh eye, the tissue is a lot softer. It's obviously more of what it would be like when it's in the animal, when it's alive. So I'm going to go ahead and just point out some of the features of the eye that we can see on the outside before I start cutting into it. So we look at this darker circle here. This is the anterior-most portion. So this is what looks out the eye, and this is actually the cornea. This particular specimen does not have an obvious conjunctiva, but the conjunctiva would be a thin layer, clear layer that goes over the cornea and actually comes up and fuses with the eyelids. And that's what prevents material, debris, whatever, from getting back behind the eye. We look at the back part and you see all this stuff hanging off the back of the eye, and almost all of this is actually adipose tissue. So the eye is surrounded by sort of this adipose padding. And if we go ahead and peel some of this adipose tissue back and look at the posterior side of the eye, you can see here some skeletal muscle all around the eye. These are the extrinsic eye muscles, and what these do is allow the eye to move around. You think about when you move your eyes to the left, you look to the right, you're using these extrinsic eye muscles to do that. And if we look in here right in the center, you can see this sort of cream-colored, almost tube-looking thing sticking out. And this right here is the optic nerve. The optic nerve is what connects essentially the eye to the brain. So all the electric impulses that come from the retina, which is inside the eye, those electric impulses will be sent through the optic nerve to the brain. Let's go ahead and look back at the front here. And so we've got the cornea here, the clear part that light actually enters into the eye from. And going back from the cornea, you can see all this white here, the white part of the eye. This is actually the sclera, which makes up the fibrous tunic, the really tough outer part of the eye that helps protect it. It actually makes the eye a fairly tough ball. So once you start to cut into it, you'll notice that this is really tough structure. It's hard to actually get into it, and there are some tricks to get into it without it coming all apart on you. Okay, so the next thing I want to do is to start to sort of clean this up. I want to get rid of a lot of this adipose tissue so we can get to the eye itself. So I'm going to go ahead and use my scissors and just go ahead and cut through. See, it's sort of rotating along as I cut the adipose tissue off. Okay, now I'm ready to cut open the eye to point out the internal structures. To do this, it can be a little bit tricky because the outer portion of the eye, the fibrous tunic, is very tough. So if you were to just take the scalpel and start poking at the eye, try to push it through the eye, you're going to put the eye under a lot of pressure, and when you finally do make a hole in it, the fluid inside the eye can actually squirt out, and who knows where it's going to squirt. So we don't want to do it that way. What we're going to do is cut a frontal section. So we're going to cut all the way around just behind the cornea, so you end up with an anterior portion and a posterior portion. Okay? And it's again right behind the cornea is where we're going to make this cut. So to do this in a controlled way, so we don't put the eye under pressure, if you take your forceps and you try to grab a little bit of the sclera with the forceps and pin it down to the dissecting tray, so that when you take the scalpel pushes on the sclera into the dissecting tray, not straight into the eye. And that way you don't put the eye under any pressure. So I'm just making a small little hole here. And you can see this hole that I've made is right here, the little black dot. See this, the dissecting probe should be able to go right in. Okay, so there's a little hole right there. Did I make it all the way through? Okay. Now what I'm going to do is use the scissors to cut all the way around to finish that frontal section, just behind the cornea. You can see the scissors go in. And as I start this, you'll see some clear fluid. Sometimes it's a little black because it has pigment mixed in. Some clear fluid pouring out. You can see a little bit pouring out there. That's the aqueous humor. The aqueous humor is the fluid that fills the anterior chamber, right underneath the cornea. It's very watery, as the name suggests. You can see more of it pouring out here. I'm just going to keep going around, all the way around. Okay, so I just completed cutting a frontal section through the eye. So I now have an anterior portion and a posterior portion. So I can go ahead and lift off the anterior portion. You can see the cornea here in the middle. And we can flip it over and we can look at it from the inside of the eye. And now this center portion that looks clear, that's actually the cornea. We're looking through the cornea from the inside of the eye. And this black ring around it is the iris. And the hole in the middle that allows us to see the cornea is actually the pupil. And the iris is part of the vascular tunic. And also attached to that would be then the ciliary body, which helps support the lens. Okay, so we look at the posterior portion of the eye, and right in the center, we can see the actual lens is floating in the vitreous humor. So I'm going to go ahead and pour that out. You want to be careful with the lens. If you put too much pressure on it, you'll actually crush the lens, and then you won't have much to look at. So you could see the lens here, and attached to it still is some of that vitreous humor. The vitreous humor is much thicker, almost like a jelly, compared to the aqueous humor that was in the anterior chamber. So let's go ahead and look inside this posterior chamber. And right in the middle, there's this sort of light brown material. It looks like it's sort of floating in there, almost like it's completely separated from the eye itself. In fact, most of it is. This is the retina. And as soon as you cut into the eyeball, the retina actually detaches from the wall of the eye. But there's one part where it's still attached, and you can see it right in this region right here at the end of the probe is where it's attached. And that portion is actually the very beginning of the optic nerve. So I'm going to go ahead and flip it over and show you that optic nerve on the back again. And you can see the optic nerve here, this sort of cream white, and that lines up perfectly with where the retina is attached to the eye. So that's the beginning of the optic nerve, which is called the optic disc. The optic disc is what's responsible for the blind spot. Remember, the blind spot is where there are no photoreceptors for detecting light. So any light that hits the blind spot, you're not aware of in that particular eye. And the rest of this, again, is the retina, and that's where the photoreceptors are, the retina. Another point to show you is the dark color that's in here, lining the inside of the chamber. This is the choroid. And in our eyes, it's a complete dark pigmented layer. And what this does is it attenuates any light that actually passes through the retina and does not reach a photoreceptor. So if any light passes the photoreceptors, you don't want them to bounce off and potentially hit another photoreceptor. That'll provide for blurry vision. But in the cow eye, there's actually this sort of iridescent blue-colored layer. This is a reflective layer, and this can be exposed or covered depending on the light conditions. So in low light conditions, like at night, this reflective layer can be exposed so that any light that passes through the photoreceptors and doesn't stimulate them gets a second chance at stimulating photoreceptors. So although you get slightly blurry vision, you can still see. But we do not have this reflective layer in our eyes. Okay, now let's go ahead and take a look at the lens. So I'm going to very gently try to get some of this vitreous humor off the lens without destroying the lens itself because it's very delicate. And when you get the lens out, make sure you hold it up to the light and look through the lens to see if you can see an image through it. It's a little opaque now, but when it was living, it was much clearer. And you can actually see through it, and you could see that everything appears upside down and backwards. And that's just how our eyes work. Right? Images that go through are upside down and backwards, and it's the brain that is able to flip this image so that we perceive things oriented correctly. And this also, it's just like how microscopes work. Images are upside down and backwards. So that's the lens there.